Let me get rid of this. Today is Wednesday, that is true, but it is not March 31st. We do not go back in time. Today is the 7th. What we're looking at today are karyotypes to examine inheritance and chromosomal disorders. So we're wrapping up our last little bit of genetics here. We had our mock last week. For the most part, I am pleased with the results. I think a lot of you are going to do really well on the real thing. My hope is also that the real thing is a little bit easier than that test was. So that test was, I think, from 2 or 3 years ago, and I found it to be particularly difficult. So I'm hoping that your guys' test coming up is a little bit easier than that one was. That being said, we are now on a path to the star. We are less than, let's see, 3 weeks, right? 3 or 4 weeks, probably less, heading into the star. So you had your English star already. Biology, I'm pretty sure, is going to be next. Our biology exam is going to be on May 13th. So the star will be on May 13th. Today is the 7th. So as far as dates, what that means is that we are looking at 1, 2, 3, 4. 4 weeks away from the star. Now we're going to take a few days to wrap up our unit on genetics. We're going to talk about biotechnology, karyotypes, gel electrophoresis, and GMOs. And then from there, we'll be done. Like I said, we'll have our big unit test coming up here pretty quick. My assumption will be that that'll be next week. Maybe. We'll see. And then we'll have our star review, and then we'll have our star. So we're almost there, guys and girls. Okay, let's go ahead and get to our exit ticket. Oof, I'm jumping way ahead. Into our class kick. The code is already in the chat box. I'm going to give you 3 minutes to work on this do now. Here we go. Make sure you give me a thumbs up when you are finished with your do now. I'm hoping it does not take you 3 minutes. Since we're getting ready for the star, we're going to go all the way back to the beginning. Do a little bit of review. That way when we do our star review after our unit exam, we can definitely hit the ground running. Thank you, Josiah. Thank you, Zaria. Yeah, please, I hope this is not taking 3 minutes. 3 minutes, but I hope that it takes you a lot less. This is one of those easy star questions. You should look at this and say, thank goodness, this is an easy star question. Thanks, Hugo. Like I said, I hope you guys had a great, relaxing evening after such a long day. Hope that you guys are ready to finish the week strong. It's already Wednesday. We just got like 2 days left, and then we're back to the weekend. And these next 5 or so weeks are going to go pretty quick. We got a little less than 30 seconds left. Thank y'all for giving me a thumbs up when you're finished with the exit with the do now. Even though I'm encouraging you guys to get lots of rest, I think it's pretty clear at this point that I am not getting enough rest. Thank you, Terrell. Okay, let's go ahead and transition over into class kick. Okay, so here we see our practice star question. Like I said, it is a very easy question. Some students used information they gathered from lab investigations to prepare a table. They entered the table in their lab notebooks. We got cell 1 and cell 2. Cell 1 looks like it's pretty small, less than 5 micrometers. Doesn't have a nucleus. Does not have membrane-bound organelles. Has circular DNA. Now this last one you might not know 100%, but I'll tell you that that is called a plasmid. So we'll come back to that. Cell 2 is larger than 10 micrometers, has a membrane-bound nucleus, has membrane-bound organelles, has linear DNA. Now, I'm going to say that maybe you don't know about circular or linear DNA, so you're not 100% sure about that. You're not even really sure about the size either, because what is 5 and 10 micrometers? The real takeaway is that one is smaller than the other. So this one's bigger than this one. But what I will say is that this should definitely give it away. Does not have a nucleus, has a nucleus. Does not have membrane-bound organelles, has membrane-bound organelles. But again, the nucleus is going to be the biggest giveaway here as far as what we're talking about. So you go down to your answer choices. We know that if we're dealing with nucleus and no nucleus, we're talking about prokaryotic and eukaryotic. Now, really quickly, you can definitely get rid of H and J. The reason you get rid of H and J so quick is because it's saying that both cell 1 and cell 2 are the same. Clearly, they are not, right? We have two different cells here. They have different characteristics. They're not going to be the same thing. So, easy, get rid of those two. Now you're down to 50-50. So, eukaryotes, prokaryotes. In the chat box, let me know, what is cell 1? Cell 1 does not have a nucleus. In the chat box, tell me, is that eukaryotic or prokaryotic? Very good. Very good, you guys. Very good. I'm looking for as close to 100% as we can get. Thank you, thank you. Yeah, you can put pro. That's fine. I understand what that means. One more. Awesome. Thank you, guys. Very good. Prokaryote. Now, this is not a spelling test, so again, it doesn't matter. That's why I think you can also put pro. I get what you mean. So, prokaryote. If we know then that cell 1 is a prokaryote, get rid of that, which means that G is your answer. That's simple. Again, my hope is that the questions on the star are going to be like this because these are nice and easy and zero stress, right? So like I said before, we're going back to the beginning as we prepare to review all of our biology content. We still have a couple of days, maybe even a full week of genetics to finish, specifically biotechnology. But after that, we're going to be heading into our star review. Okay, any questions about that? Hopefully not, because again, it's pretty basic stuff. Great. Now, we're heading into karyotypes. So if you don't know what karyotype is, there it is right there. We can see all of our homologous chromosomes lined up. Nice and pretty. So we're going to watch a video about karyotypes so we can answer some questions about what they are, and then we're going to look at some karyotypes.
I've seen too many pictures of chromosomes. Well, diagrams, really. Diagrams are awesome, but I've learned I need to be careful about misconceptions that can come with them. See, in diagrams, I've seen chromosomes typically drawn in an X shape. So when I was younger, I thought, okay, all chromosomes are an X shape, which is really confusing when I heard people could have two X chromosomes or an X and a Y chromosome, because then I started to wonder, why are these chromosomes resembling the letters of the alphabet? And then sometimes they look like single stick pieces and not X shapes, and I wondered what is going on with these things. I also thought that if humans have 46 chromosomes, then they're in there somewhere deep inside the body, that if you need to analyze them, you'd have to find them and make sure not to do anything to them because you only have 46 of them. Well, these are some misconceptions I had. We're hoping to clear up a little bit about chromosomes. Now, our illustrations are not scientific, obviously, but they can help with our explanations, which are. While you can find chromosomes in bacteria where the chromosomes tend to have circular shapes, we are going to focus on eukaryote chromosomes. Specifically, we're going to focus on human chromosomes. Chromosomes are made of chromatin, and chromatin consists of DNA and protein. A chromosome itself is really intense packaging. It starts with DNA being wound around proteins called histones. This forms nucleosomes, which are often considered to look like beads. More and more condensing packaging levels occur. We highly encourage you to explore this until it results into the chromosome level. But why all this packing? Well, in your cells, you have a lot of DNA and it has to fit inside a nucleus. Another reason it's also really important for when you make more cells, like in the process of mitosis. But in mitosis, you have to be able to move DNA into new daughter cells. You have so much DNA that these highly packed chromosome units make it easier to do so. By the way, it's not always packed up so tightly. For example, in DNA replication, DNA typically needs to be unwound. There are a variety of factors that also influence how tightly packed chromatin is. Let's get kind of a general idea about how human chromosomes may be represented. Human chromosomes can be like this as a single component, or they can be replicated, like this. In our chromosome counting video, we mentioned when chromosomes are duplicated during interphase, the newly made chromosome is still bound here in this region called the centromere. By the way, this region does not technically have to be in the center of the chromosome. Because of it being joined here, we still count this as one chromosome unit. But that can be kind of confusing. We say that there are two sister chromatids. So one chromosome here, it duplicates, and now, while this is still attached, it's still one chromosome. So in humans, you have 46 chromosomes. In interphase, they are duplicated. There are still 46 chromosomes here, but that's 92 chromatids in the picture. During a stage of mitosis, those 92 chromatids get pulled apart, so each cell will have 46 chromosomes. Now, just a side note, while most of your body cells have 46 chromosomes, it's important to remember that certain genes on the chromosome may be turned on or off by a variety of factors. For example, you wouldn't want your eye cells actively using a gene to produce stomach enzymes. That'd be a problem. So, would you ever get a chance to see all 46 of your chromosomes clearly? Well, you could in something called a karyotype, an image of all your chromosomes. It's possible to have a karyotype made as part of some types of medical screenings. In a karyotype, the chromosomes are stained and visible. The chromosomes from a karyotype are typically from a cell that is in a mitosis stage, typically metaphase, as chromosomes are condensed and thick. A karyotype during interphase would be a challenge because you don't have that condensed DNA packing. Now, something confusing to clarify. When searching for karyotypes, sometimes it'll be represented kind of like this diagram, and sometimes you see something like this. And while both show 46 chromosomes, the number of chromatids in the two images here seem different, so what's going on? Now, while the mitosis stage is anaphase or telophase would be a time when each chromosome would have a single chromatid, those phases are generally not when you do a typical karyotype. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, the chromosome would have two joined sister chromatids. It's just that many times the joined sister chromatids of the chromosome can be so close together that to an untrained eye, it might be difficult to see that each chromosome in the karyotype technically has two sister chromatids. When arranging the karyotype, the chromosomes are arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and in each homologous pair, you receive one chromosome from one parent and one chromosome from the other parent. 23 pairs. 22 of the pairs, 44 of the chromosomes, are called autosomes. That means they are not related to your biological sex. They may have genes related to eye color or height or hair texture. The last two chromosomes are called sex chromosomes because they determine biological sex. Females have two X chromosomes, males have one X and one Y chromosome. Oh, and just a side note, the name X and the name Y have nothing to do with the shape of them. The reason is an interesting topic to look up though. So 23 of these chromosomes are from the father. from a sperm cell, which is a gamete. Unlike body cells, gametes have half the number of chromosomes as body cells. Sperm cells are considered haploid because they only have one set of chromosomes. The other 23 of the chromosomes came from the mother, contained in an egg, which is also a gamete. An egg cell is haploid because, like the sperm cell, it contains only one set of chromosomes. When a sperm and egg cell combine, they form a fertilized egg, known as a zygote. The resulting cell is diploid, as it contains two sets of chromosomes, and it will divide to continue to form more diploid cells. You are a diploid organism. Now, if you look at the sex chromosomes, you know that mothers can only give an X chromosome, because that's the only type of sex chromosome they have. But fathers could give an X, the baby would be XX, female, or a Y, the baby is XY, male. So fathers determine the biological sex. So why do we care about karyotypes or learning about chromosomes? Well, one reason is it can really help when we are trying to understand genetic disorders. A karyotype can reveal missing chromosomes or extra chromosomes at specific locations. Do you have an interest in a career that involves this area of study? The field of genetics continues to expand, and the career of a genetic counselor may be something to read up on. Well, that's it for the Amoeba Sisters, and remind you to stay curious.
Okay, so the simple explanation is that your chromosomes need to condense during division, right? We learned about that during mitosis and meiosis. So they line up along the center of the cell. Remember, like in metaphase, that's when these cells are kind of like taken apart and then arranged in a specific order. So this is not how your chromosomes appear in your cells. These are kind of artificially done this way. So the homologous pairs, remember we got one from mom, one from dad, are kind of lined up and then they are stained so they can kind of see the differences between them. Okay, so if I could get a volunteer to read these two paragraphs for me, please.
I'll read it.
Thank you, Miguel.
Do I read the introduction?
Mm-hmm.
Okay. Several human genetic disorders are caused by extra, missing, or damaged chromosomes. In order to study these disorders, cells from a person are grown with the chemical that stops cell division at a metaphase stage. During metaphase, a chromosome exists at two chromatids attached at the centromere. The cells are stained to reveal bonding patterns and placed on glass slides. The chromosomes are observed under the microscope, where they are counted, checked for abnormalities, and photographed. The photograph is then enlarged, and then um, and the images of the chromosomes are individually cut out. The chromosomes are identified and arranged in homologous pairs. The arrangement of homologous pairs is called a karyotype. In this investigation, you will use a sketch of chromosomes to make a karyotype. You will also examine the karyotype to determine the presence of any chromosomal abnormalities.
Thank you so much, Miguel. Actually, before I say that, how about thank you in Korean?
감사합니다.
감사합니다. Sorry. Okay, so thank you. Now we're going to make karyotypes probably tomorrow, maybe even Friday. For now, we're looking at the basics and how to do it. And because I'm already running out of time, I think I might even skip a lot of these and get to the good stuff first. Okay, so let's look at this. Observe the normal human karyotype in figure 1, which is what we've been looking at. Notice that the two sex chromosomes, pair number 23, all the way down here, do not look alike. They are different because this karyotype is of a male, and a male has an X and a Y chromosome. So what does that mean? That means that when you're looking at these chromosomes, you have an X and you have a Y. Now I'll go ahead and tell you that the Y is going to be the short one. So this little tiny thing is the Y chromosome. So what that means is evolutionary scientists think that what happened was a long time ago, like way, way before even people were around, there were X chromosomes, and the X was just kind of like the dominant chromosome. Then over time, you had this deleterious effect. Like pieces of it were deleted, and it got like weird and mutated. And that's where you get the Y chromosome. So men are like some kind of weird derivation of the X chromosome. So the X chromosome has all the good stuff, like hair color, eye color, all that stuff. The Y chromosome has all these weird male traits. So, like, male pattern baldness, ear hair, all these weird things that you think of as, being typical male traits are carried on the Y chromosome. So it's pretty strange. But when you're looking at a karyotype to determine genetic sex, you'll see a difference in sizes. That's how you know you're dealing with a man. If it's going to be a woman, then you'll see two of the same ones, right? You'll see XX. Now, something I wanted to show you that I haven't really covered that much when we were talking about Punnett squares is when we're dealing with, genetic sex of a kid. So we know that when you have a boy or a girl, it's going to be 50-50. As it was mentioned in the video, dad has two chromosomes, right? He's got the X chromosome and the Y chromosome. Mom, actually it looks like an X. Mom has two X's, right? So mom is only going to have the X chromosome donated to the kids. So you bring down dad's X, then you get 50% girl. You bring mom's X over, you bring down dad's Y, there's a boy, and there's your boy. So that's how you get that 50-50, right? You got 50%, 50% because of the chromosomes of mom and dad. So depending on what happens during independent assortment, like we saw with our dihybrid crosses, will determine what kind of genetic sex the child will have. Now, the different conversation about gender is more complicated, and I do apologize if I mention gender interchangeably with sex. That's just how I learned it and how I was raised. But I will admit that in this, the world that we live in right now, it's not always so cut and dry. And we understand that there are a lot of other things that can happen along the way. So, genotypically, what you're going to see is XX or XY. But there are other possibilities. You could have like two X's and a Y. You could have one X and two Ys, or triple X's. So there a lot of these different combinations are possible, and that can lead to some tricky stuff when it comes to gender. Not only that, but even in the way that a simple XX or XY are going to be expressed can lead to some interesting evolutions. So the discussion about gender is very complex and is definitely not something that's covered in your high school biology course. If you are curious, I would definitely recommend that you do some research on your own to kind of learn more about that, because it is very interesting that we don't necessarily live in a world where it's just, male, female. But that's beyond the scope of this class, so we're not going to worry about that. Alrighty, so let's go ahead and look at some examples. So we've talked about what they are. We've got some basic understandings of how they work. What we're going to do now then is look at some examples of some action. So the square here on the right is the normal one. So what we're looking here are for chromosomal abnormalities. So we got 1 through 23. So 23 pairs of 46 chromosomes, until you notice something down here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. Now, even though there are 46 chromosomes, I hope that you noticed there were two pretty big differences. There's a difference here. There's a difference here. So we have a single chromosome at chromosome number 7, and we have 3 chromosomes, or actually we'll say 3 sex chromosomes. Specifically, we have 2 X's and a Y. So here we can notice that there are differences in this person, right? This is a normal person that's, out in the world doing whatever, and they can have these genetic abnormalities. Just because you have them doesn't necessarily mean that you're like gonna be some monster. It just means that you're missing some stuff, maybe have some extra stuff, and it could lead to some interesting phenotypes. But we're looking at right now just the abnormalities. We'll talk about specifics here in a little bit. All right. Now we're looking at figure 4. I'm sorry, we're looking at figure 5. What I want you to do in the chat box, let me know if you see a mutation or something interesting. Remember, we're comparing it to our normal one up here. Okay, normal in quotation marks. In our genotypically normal. Go ahead, let me know in the chat box, what chromosome number do you notice something that is different from our baseline? Again, we have 23 pairs of chromosomes. 22 of them are called autosomes. That just means that they are not involved in gender or sex. The 23rd chromosome is the one that's involved in determining that. All right, I'm seeing the 23 in the chat box. Anybody else? Let me know. What number chromosome do you notice some kind of interesting difference between figure 5 and the baseline? Very good, you guys. I'm seeing 23. Now again, last one we saw, we had 2 X's and a Y. Here we see a single sex chromosome at chromosome number 23, which are your sex chromosomes. Now, a single X will usually mean that this is going to be female. Because again, male is going to require that Y chromosome to display some of those the male traits. So if you only have that single X, the body is going to default to female. Now, there's a really interesting thing that we don't have time to talk about, but that we definitely would if you were in AP Bio, called X chromosome inactivation. What that means is whenever you're dealing with those chromosomes, we saw with the Punnett square that you're getting both of those X's from mom. It's also possible to get one from dad. What it means is that you're going to have one of those being inactivated. So one of them could be mom's, one of them could be dad's being inactivated. So only one is ever going to be expressed. It becomes really, really interesting when you start looking at twins and sisters. But like I said, that's more of an AP Bio concept that's never going to be talked about on the star. It just means that since women have 2 X's, some cells have some X's turned on, some of them are turned off. Pretty interesting. X cell inactivation. Awesome. Okay, we're going to try something a little bit interactive. Now, there is a link here that says patient histories. What we're going to do is we're going to look at 3 unique patients, patient A, B, and C. So what you want to do, well, first let me know in the chat box if it works. It works on my computer, but I know that sometimes the links don't work on Elle's computer. So real quick, give me a thumbs up in the chat box if you're able to open up this link and see the different patients. Great, great. I'm seeing some thumbs up. I like it. Good, because this is a cool kind of activity. All right, so we're going to go with patient A first. The first thing that this activity wants you to do is to match up the chromosomes. So you're kind of doing the same thing the scientists do. As a reminder, what's happening is that during metaphase, when your chromosomes are condensed, they appear as these bands, right? So what you're trying to do here is you're trying to match up the chromosomes. So here you see that there's the chromosome, and you're going to click on the number that it matches up with. So this, you could count the bands that are stained to, I think, this one. This one looks pretty small. I think it's probably going to be down here on 16. We got this one. Looks like it's probably going to be number 12. And then finally, we got this one. And there we go. So this is what the normal karyotype should look like. Again, normal going to be in quotation marks because defining normal when it comes to genetics can be dangerous. So genotypically, this is what you're going to see. Now, we have an extra chromosome. Where is it going to go? It's very small. I really only kind of see one biggish band. So you look down here, these are your small ones. You kind of look at your banding patterns, and it looks like this one matches this one. There you go. So we've completed patient A's karyotype. Now, let's make a diagnosis. Next step is to either diagnose or rule out a chromosomal abnormality. Now we know that there is an extra chromosome somewhere in this karyotype. Okay, so we look again at our karyotype. The extra one that we put was here at 21. So the 21st chromosome has an extra one. Now, you might know that this is called trisomy 21. Now, if you don't know what that means, don't worry, we'll talk about it here in a second. So let's get into who this patient is. Patient A is a nearly full-term fetus of a 40-year-old female. Chromosomes were obtained from fetal epithelial cells acquired through amniocentesis. So what happened was we got a pregnant lady. We got a sample of the skin tissue from the fetus growing inside her, and we are now looking at the chromosomes. This is actually something that's done fairly commonly when you're dealing with pregnancies. So for those of you who don't know, I had a baby recently. Well, I guess, 2 years ago. And so what happens is whenever you're dealing with some kind of complication, so for example, my wife was having a lot of trouble, you try and see if the baby's okay. So you can run tests on the baby. One of the tests you can do is looking at the genes to see if there's any kind of abnormalities with the baby. So this one was run, and we see that there was an abnormality with chromosome number 21. So let's go up here to our diagnosis. We have that trisomy there at 13. So this is going to be an extra chromosome at 13. Now, what does this mean? Unfortunately, that does mean that this child has a chromosomal mutation, specifically that extra chromosome at 21. The way this can happen is that during development, we can have like what's called a non-disjunction. So you know how the cells are supposed to separate and everything's supposed to be perfect? Well, sometimes it's not perfect and you can have some stickiness. So here we have some kind of duplication of that 21st chromosome leading to this disorder. Now, many of you might recognize trisomy 21. The more common explanation for this is called Down syndrome. Ooh, why did I say trisomy 13? Down syndrome. So trisomy 21, extra chromosome 21, right? So this 21st chromosome is called Down syndrome. So what that means is that whenever this baby is born, the baby will be healthy, but it does mean that it's going to have some learning disabilities for the most part. Now, that doesn't mean that if you're born with Down syndrome, it's some kind of death sentence. That's not true. It just means that the baby's going to need a little bit more extra love and support. That's all. But it's showing you the amount of power that your DNA has over who you are as a person. So when you think about, the DNA being your hair color, your eye color, your personality, the way you think, the way you feel, how you're able to eat food, how you're able to move around, there is so much delicate balance to how that all works. It's kind of a miracle that it happens all the time so perfectly. So there is, some things that can develop that can lead to somebody having these chromosomal disorders, in which case their lives will be impacted by that. Okay, so very good. That was patient A. Let's go back to our class cake. So patient A, the summary is basically that we had a fetus, let's make it a little smaller, of a 40-year-old woman. Now, the reason that's important, not that any of you are family planning, as you get older, what happens with women's eggs is that they are more prone to these types of mutations. So you might hear that as you get older, especially you ladies, that it's important that if you want to avoid a lot of these situations, the best thing you can do is try and have your kids early. And that doesn't mean like 14. It just means that as you get older, these things are going to happen more frequently. So especially at 40 and really even above 35, it's more or less common now to be having kids at 30. But even that is probably the upper limit because everything after that, you're more prone to a lot of these genetic disorders, specifically trisomy 21. So this woman is, pretty old. I guess, I mean, she's not old, but 40 is a little old to be having babies. And so they are more prone to having these types of mutations. That doesn't mean it's not possible to have a child with a completely normal karyotype. It just means that it's more likely that you're going to have a situation like this happen. Okay, we counted our chromosomes and we counted 46 normally, but here we have an extra one, so it's going to be 47 chromosomes because of that extra one on 21. Now, the last set of chromosomes are the sex chromosomes. You got two the same, you got a female, you got one big, one small, that's going to be male. What sex is this baby going to be? So in the chat box, let me know, are we dealing with a boy or are we dealing with a girl? Again, the sex chromosomes are determined by this 23rd pair. You got two of the same, you're dealing with two X's, which is a girl. You got two different ones, you're dealing with a boy and a boy, like XY. So in the chat box, let me know, what are we dealing with? Thank you, Hugo. Josiah excellently put. A female, since the chromosomes are the same, right? So you see the banding patterns, the size, everything points to it being two X's, in which case it's going to be female. Now, just to reiterate the point, male and female in today's day and age, a little bit more complicated than it used to be. So right now we're just dealing with genotypically. We know that the extra chromosome is going to be a chromosome number 21, which is what led to the disorder. Specifically, the disorder is going to be Down syndrome. Now, again, I apologize for using the word normal. It's just, it's hard to say the correct words all the time. So the reason it's so dangerous to say normal is because you don't want to label somebody with Down syndrome as being abnormal, right? Like I said, they can still function, they can still live a wonderful, happy life. And so to just say that their genes are not normal is, not only not politically correct, it's just not true. It's also led to a lot of marginalization for people with these types of disorders. Even calling them disorders could be labeled politically incorrect. A big point of contention for this is usually drawing comparisons to like fascism or the Nazis back in the 40s. So the first thing that people think of is usually the Jews, right? The Holocaust, the camps that were built to eliminate the Jews. So for those of you who might not know your history super well, what happened was back in the 40s in Germany, they pretty much blamed all their problems on Jews, similar to how with certain administrations recently, blaming all of our problems on immigrants. The same thing was happening back in Germany. So there was a fascist movement, which means that people were really proud of their country and they wanted people to be, proud of their country. Part of that was because World War One had ended recently and the country was very, very poor. They had a lot of problems because Germany were the bad guys back in World War One as well. So they were trying to rebuild their country. A big part of that meant that they wanted to have a lot of pride in who they were, which kind of led to this uprising of people who were, putting white supremacy kind of, on top. So, along with blaming all their problems on a specific race, Hitler also wanted to make sure that he was, quote unquote, purifying the race and getting rid of a lot of these genetic abnormalities. So genetics was still relatively new back then, but they knew enough to figure out when people had these mutations that they would have certain disorders like Down syndrome. So Hitler not only was getting rid of Jews, he was also eliminating these undesirable traits from the gene pool. So you might have heard about the Aryan race, which is like, white, blonde hair, blue eyes, that sort of thing. He was trying to genetically purify the Germans to have a certain type of genetic trait. So he was trying to get rid of all the people who didn't have that. Now, the big things that he did, like I said, with the Jewish population was to exterminate them. You also had other ethnic groups like gypsies. He was also trying to get rid of them. And of course, people with Down syndrome and other genetic disorders. You don't hear about it as much, but that was also part of the plan. So as we get into biotechnology, I want this to be a big realization for you that there's a lot of positives that come with this, but there's also a lot of negatives. So history kind of points to, some dangerous methodologies when it comes to genetics. It's a really cool tool to help us understand more about ourselves and how we function, but it could also be used by people who are not necessarily out there to respect everyone. So, like I said, a big point of the Nazis were trying to purify the race. Like they were trying to say, okay, these genes, these alleles are desirable. These are the ones that are quote unquote normal. This is what we want everybody to be. Everybody who does not meet the standard then will become abnormal. So we're definitely going to talk more about this the next couple of days. Not necessarily the Holocaust, but the ethics of genetics. So we've learned kind of the basics. You're able to recognize, chromosomes and genes and all that. That's only part of the equation. It's like saying that, um, hamburgers taste good. Hamburgers do taste good. Hamburgers are wonderful. But there's a whole other history to hamburgers that you have to be aware of, right? There's processing plants, there's cows, there's these other living organisms that are actually dying to give you food. The food is great, the food is wonderful, but it comes at a price, right? Same with genetics. So there are a lot of things that are really cool about genetics, but there are a lot of things that are unethical. Now, I'm not saying that it's bad to study science. That's not what I'm saying at all. Science is a tool. The problem is the way that the tool is going to be used. So I think it's really important for you to learn how these tools are used and what they mean. But as far as how the tools are going to be used in society, especially in the past and looking to the future, are going to become very important. I think that we're on the precipice of a lot of really interesting things with genetics. Specifically, we're going to talk more about it, but there's something called PCR, which is basically a way for you to amplify genes. So I mentioned it a while ago when we were talking about DNA. Remember that DNA is common to all organisms, from bats to whales to spiders. Everything that's alive has DNA. We can now modify DNA pretty much at will. The problem is that we don't really know a whole lot about it. Think of it this way. Think of you taking your first trip abroad, like you want to go to Japan, right? Now you get on a plane, you go to Japan, you get there, wow, it's cool. You see all these new sights, these new sounds. The problem is you don't really know how to interact with any of these people, right? You don't speak the language. You don't know how to get around. So even though you can, get there and you can do stuff, you're kind of just doing it without a guide, without any kind of instructions. And that's really where we are right now with genetics. We have a really good understanding of how things work, like you probably know how to, live and breathe. But when it comes to this whole new aspect of genetics, as far as we can modify genes now. We can insert genes and control genes. Like if you want to make a unicorn, well, hey, let's take a tusk gene from an elephant, combine it with a horse, and maybe we can get a horse with a horn, right? But the problem is that we don't really know 100% how that's going to work. Now, that's where you get into this ethics thing again, because where do you draw the line between something being ethical and unethical? Like, let's say that you wanted to make this unicorn. Is it ethical to create a horse with this weird tusk gene? Because you're essentially controlling the offspring without their consent, right? It could be seen as, cruel because we don't know if we're going to make a unicorn. For all we know, that tusk is going to form in the middle of their chest cavity, and suddenly you have this weird bone tissue inside these organs, and you're going to make a horse that can't live. So you're essentially creating an organism to die, and that's just animals. Imagine that with people. And then, of course, like I mentioned with the Holocaust, with Hitler trying to say, okay, these are the genes that I want, these are the genes that will continue, we are going to be pretty close to that happening on a regular everyday scale when it comes to deciding the genetic fate of children. Like right here, you can see that we know that this child, if it was going to be born, because right now it's just a fetus in this 40-year-old woman, we know that this child is going to have Down syndrome. There might be, a lot of impulses that come with that. The one that we've already had already and that we deal with, and depending on your own political or religious beliefs, is a very serious discussion about whether or not even to continue with the pregnancy. Do you terminate the pregnancy if you know that you're going to give birth to a child who's going to be struggling with this disorder? Right? So that's already number one. That's something that we've been dealing with for a long time already. But now, number two, you can hypothetically change the child's DNA to not have this disorder. Do you do that? Do you not do that? Will messing with the child's genome significantly, you know, mess up this child further? If you are controlling this child's genotype, are you saying that this is a mistake that shouldn't happen? Is this something that the child would be unable to live with? So, and then of course experimenting on babies, like, is that something that you should be doing anyway? So there's all these extra questions. So I want you guys to be really thinking about that as we move forward with biotechnology and really almost everything that we've learned this year in biology. There is a whole other aspect to everything that we've learned in this class. And not just this class, but really all the other classes that you've been taking. I'm showing you how the world works. Everything that we're learning in this class has to do with, biology. Bio meaning life, low G study of. So when you're looking at these things, remember that there is a separate whole piece to this. You're learning what they are, but then you're going to learn about what to do with that knowledge. Kind of like Jurassic Park, right? When uh, Ian Malcolm says, they were so busy thinking about the things that they could do, they weren't thinking about, what they should do. So I think learning in itself is not a bad thing, right? It's great to learn more things. The problem is going to be how we move forward with this knowledge. And that's something that your generation and my generation are really going to have to start grappling with as we discover more and more tools about how to do this. Tomorrow or Friday, we're going to watch a video about biotechnologies as far as how cheap it's gotten. In the past, we've learned a lot about this, but it's been really, really hard to do, really expensive to do. It was time consuming. We weren't really sure how to do it. But now, 2021, right now, you can modify genes for less than $20. You can go online, you can buy kits, and you can basically mess with your own DNA right now. And that's happening currently. There's a group called biohackers, and what they do is they're trying to modify their own DNA. So they've gotten these tools, they're using them on themselves, and they don't know what the outcome is going to be. The dangerous thing is that if they are messing with their genome and they start to reproduce, are these new genomes going to be introduced into the human population? What does that mean for the future of humanity? Will we still even be humans if it's happening on that big of a scale? Because people want to, maybe they want to have bigger muscles, or they want to be smarter, or they want to grow a third arm, or whatever. I mean, who knows, right? If you're messing with your genes, you are radically influencing the future evolution of the human species. So this is a culmination of everything you guys have learned. Evolution, ecology, right? The ecological relationships. We exist in balance with each other and with the world. Cells, genetics, right? How are these things going to affect the individual? So I know that I'm rambling, but I really think that this is important as we head into the end of this year to really start thinking critically about everything that you've learned. I know the point of this class is to get ready for the star, but I really want you to be thinking about this as you as a human being. As you're starting to get older, I want you to really think about what the world is and what it means to you and what the world of you is like inside of you. Okay? All right. Again, I didn't mean to go on a giant rant. We're going to work on this again tomorrow because we clearly did not finish today. I just think that it's really important that you guys understand genetics. Like, it's not just Punnett squares. It's not just karyotypes. It's what does all this stuff mean for you, for the world? And that's where the questions become interesting. Even if you don't think that it's going to affect you, this is going to be your future. Okay, like I said, rant over. I'm putting the exit ticket into the chat box. Like I said, we're talking about biotechnology for the next couple of days. We'll wrap up our unit talking about this. We're going to have more ethical talks, and really, I want you guys to be involved. So this one was just me talking because you guys are just being introduced to this at this point. But we're going to take time to really think about what we feel about the future of genetics, about what it means to have, these new modified populations. Like, let's say, for example, that, My daughter didn't have anything done, but her generation are going to be the ones that, you could. What if we modified these babies' genes? Suddenly, you now, 20 years later, you are going to be this population who did not have their genes modified. And you're going to have this new population of kids who have their genes modified. What if they're smarter than you? What if they decide that your genes are not good? Right? Then it opens up a whole new can of worms. So I really want you guys to be thinking about, again, not only you, not only this class, but the future and how it impacts our world. Okay, we got about 10 minutes left. The exit ticket is pretty simple. I wanted to keep it simple because I know that we had our STAR test yesterday and you guys are probably still recovering. That's why I was trying to keep it, even though we are talking about some heavy philosophical things, I was hoping that, this class would be relatively interesting to listen to. Okay, so there's a link for the exit ticket. Like I said, not too difficult. The end of the 6 weeks is probably, or not 6 weeks, end of the 9 weeks is coming here pretty quickly. So make sure that you are completing every assignment because we are going to have, another shutdown, I think even next week. Next week we have our AP Human Geography mock. So that we're going to lose a whole another day, right? And then of course we're going to have our unit exam coming up pretty soon. And then we're going to be having stars. So there's more time, or there's less time now than there has been in the past. So it's really important to get these done. Now, like I said, we are going to finish this assignment tomorrow. So tomorrow we're going to be looking at some more karyotypes. We're going to revisit this, so don't feel like, you got cheated. Okay. Oh, Caleb, didn't even notice you. All right, I'm going to stop the recording. I will put on some music so we can work on our exit ticket.